right, thanks.
All right. So we're gonna talk since we're talking about Eli, anyways. You and our paper with Eli and Tank on this. I guess half of the data also came from the Tank data set, I think. About some idea of explaining the response properties of grid cells in 1D environments. And sort of the main hypothesis that they're testing in this paper is the idea that you can explain the 1D responses, which are quite different from the 2D responses, and that they have varying peak amplitudes and they're not periodically arranged, multiple response fields, and mean spacing between fields that is larger than 2D arrangements, but that you might still be able to explain it as slices through a 2D lattice. Yes.
Maybe too early. How do they, like in the 1D environment, are these animals only ever subjected to a 1D environment their entire life?
No. No. No.
Okay.
In fact, these are very specifically animals that behave in both contexts. And the same tetrad recordings.
But at some point in time before the experiments, they were like existing in a 3D world.
Yeah, you're not born into a 1D world. Yeah, no.
It's almost like, you could be in, you might have one model for a room or an office, but then if you're going down a hallway and you learn that hallway, you may not think of it as much as two-dimensional or one-dimensional. So what is your mental model for that hallway, that kind of stuff. So right. You feel like you live in a 2 or 3D world, but sometimes you live in a 2D world or a 1D world.
Right.
Yeah. I think it's also important to know these are not, most of these lab mice and rats are, it's not like they grow up in a very rich, stimulating environment. They're often in these very restricted little cages, and they might have a few toys to play with, but they don't have the sort of wild type experience.
And in these cases, actually two different kinds of linear tracks, both the sort of treadmill variety, physical track, as well as a virtual track. So I guess, VR created the stuff that David Tank pioneered.
Before you go forward here, I want to make sure that this is what I think I'm talking about. Because I gave a talk last Monday, which most of you missed, since I was here, some of you missed, and I was talking about the idea that maybe I'm walking through the idea that a cortical column has a whole bunch of 1D grid cell modules that are aligned with mini columns. But the mini columns themselves represent 1D. And I went through, although we had no evidence for that, I led to a very nice explanation for how a bunch of things work. So there was like no empirical evidence for this, but there was a lot of theoretical bias because it explained a whole bunch of stuff.
Right.
And then you said you're going to look for the page. Is that what you're still doing here? You didn't mention that.
Yeah.
So this is not randomly, we're just looking at this. We're trying to figure out if there was, what does the literature say that might possibly be explained by the thing I talked about.
Exactly. So I wanted to
say that.
Right. Okay. Yeah. So to be clear, this comes from this discussion of Jeff's idea, and I'm essentially like revisiting a couple of different place cell papers, grid cell papers. Now to sort of see this in the context, we can find ways to affirm it or contradict it, cast doubt on it, or find interesting new perspectives to view that.
So the high level idea was that the grid cells in the columns are using like 1D representation.
The high level, we have a problem with the thousand brains theory.
You have a problem?
Yes, the problem is the thousand brains theory says, we have grid cells in each cortical column. But the grid cells to work, you have to have multiple grid cell modules. You can't just have 2 or 3, you got to have a bunch for it to really work. And we had no way of understanding where those grid cell modules are in a cortical column. In fact, even in the entorhinal cortex, it doesn't look correct. It doesn't look like there's enough modules there. And there's some other weird data that we have too. So there's sort of, and then even then, we didn't know how orientation impacts grid cell modules. We didn't have a really clear understanding of that. So the basic idea I walked through on my last Monday was that I start off by pointing out that the cortex is really just a whole bunch of mini columns. That's what it is. It's just a whole bunch of mini columns, and you group them, several hundred of them together, and you've got a regular column. And so the idea was that could there be a one-dimensional grid cell module in each mini column? And therefore, you'd have several hundred grid cell modules that are very impoverished, but on the other hand, there'd be a lot of them. Yeah. And then I walked through from that idea. Each one of these guys is intersecting some n-dimensional spaces along the paper that Mirko and Marcus worked on. And under that idea, you could how would each mini column learn what what dimensionality it is and how it would know to update itself with sensory input. There's a whole bunch of things that came out of it that sort of made sense, which we didn't really understand. But that's the basic idea. But that would be a vertical one-dimensional grid cell module, not like a horizontal two-dimensional.
And you think that it'd be a one-dimensional because it's such a thin problem?
Because it's theoretically everything, I can get a whole bunch of stuff to work out that fits.
You get 30 cells to work with.
So I guess what I'm saying is like, you wouldn't think 2D just is the limitation of the number of cells.
No, at least I can understand conceptually how a 1D grid cell module easily gets updated.
Oh, I see.
So for example, when you have an orientation, I'm sorry to just direct you, but if you have an orientation, like you have let's say you work in a 3D world, you have to have a 3D orientation, like which way you're facing. And somehow you have to take that 3D orientation and use it to decide which way to update the 3D grid cells or the multiple grid cell modules based on your movement, right? It's like, how do you do that? It's really hard. Mathematically, you can show it, but neurons is very hard. So this, what you end up here is all you have to do is each mini-column has to just know its orientation is basically a scalar value, which is the how much of that mini-column is projecting into the, where your current orientation projects into that 1D space. So each mini-column can update on its own, just like by one scalar value. And then I showed how you can actually detect that scalar value. So it's like, it's just a whole bunch of things fell together nicely.
Okay.
But there's no evidence for this.
I think that's where you're talking about the theoretical bias.
Well, it's like, yeah, theoretically, I can explain a whole bunch of stuff,
Yeah.
but there was no evidence for it. Then we had a little speculation. Could the 2D grid modules that we see in the entorhinal cortex actually be a bunch of 1D models that it look like 2D modules under certain conditions? I'm not saying that's the case,
But it sounds like that's what this paper is exactly trying to figure out.
Well, let's see where it goes.
Okay.
Right? It's
So
a lot
that's-
more interesting with a bit of
Hour
suspense.
starts now.
yeah.
Right, yeah.
You can watch
Thank
the,
you.
we did the last week's online.
Right.
Okay.
So first things first, you start with a bit of math.
Florian's a good
or like
sport.
just very broad idea. I guess you're familiar with the 2D grid cells, right? So this is how it looks like. You have in gray the outlined track of these rodents in a 2D space, and then in red, right, you put a dot in this map whenever there's a spike, and you very nicely see that from doing so, these are two different tetraodes, different channels, I guess, as you can see from these numbers here. You get these firing fields, right? So this is what you then see here in 2D, which have this very nice 2D lattice with 60 degrees. And you can obviously record from the exact same electrodes, right? Then on a linear track. And typically the behavior looks a little bit different because it's not, clearly periodic in that sense, and the amplitude tends to vary. So if you do a firing rate averaging, right, over this track, which is like 6 meters long.
G is all one-dimensional.
Yeah, exactly. So D, E, F, right? The lower part of this diagram is one-dimensional. The higher part is two-dimensional.
I don't get it. So how can you, what G, the upper G, this is Latin.
These are the same two electrodes that you saw up here in the 2D environment. Now you see them on a one-dimensional 6-meter track.
One of those is definitely more periodic than the other.
Yes,
Okay.
exactly. And you,
I want to make sure those are both 1D recordings, even though they look different.
Right.
So obviously the cells are not responding the same way.
Right. So that is just the first order observation, which makes 1D behavior interesting. And if you plot the firing rate, so of these, instead of looking at the raw spikes here, right? So if you get sort of the average over multiple laps, then you see these very nice, peaky behavior here actually for both of these tetraodes. And so one thing that one can do, of course, is take a look at how do these sort of overlap with one another. You see, for example, that the firing fields
Are you claiming
of the first.
Are you claiming the lower one there? Is that periodic? Nice.
I'm not claiming anything
You
here
said
yet.
a nice part. I would say that's not nice.
Okay, I was jumping up to see here. I'm sorry. So back to the 2D case, right? You can look at how do these firing fields like correspond to one another, and what you can see just in this example of these two tetraodes that you can very nicely explain the firing fields by sort of slightly shifting the underlying map, right? You can see that these firing fields, this is the blue one, and the upper one is the one that is drawn here in red. They kind of fit together, which suggests they're part of the same module, right? And so when you do an analysis of the Fourier components of this firing field, so that's just a 2D Fourier analysis, you very, very nicely get those three those three sort of opening angles, right, of the lattice. So that these 60-degree angles you see in the no, it's a 2D power spectral density of the Fourier transform. So you can do the same thing, obviously, in the 1D case. So now you can try to make these firing rate things overlap instead of letting these 2D fields overlap. And that, might work for some peaks, but as it turns out, it's a little bit tricky, right?
That was my observation down there.
Right. But if you take a look, if you do a Fourier analysis and you look at the frequency components in this, because of course we might see some oscillations in here, you see that both of these electrodes have the same 3 peaks, which already suggests, well, there's, very nicely 3 different.
This is a frequency of firing, but has nothing to do with the location where the animal is.
Exactly, right? So it's a spatial firing frequency. It's a spatial frequency analysis. It's just a Fourier analysis, so it's invariant to the shift to the exact position, along as we will talk about the detail.
I'm just trying to understand it. There's no point in loving photo. I don't understand it. So
right.
So I'm just point out this frequency analysis is not obvious to me how that's useful in any way to the animal. What does it mean in relationship with the animal? It's
useful because we obviously see a spatial frequency, very characteristic, very strong ones in the 2D case. And so the question is, do we also see that when we analyze the frequency spectrum of the 1D response?
If I'm understanding this correctly, even in the 2D space, I don't understand why that's valuable to the animal. That's just the frequency firing rate of these cells.
I think it's not a question. It's just they're trying to understand the characteristics of the fire.
Just arguing.
Just saying it's at the same spatial scale and same orientation.
Okay, all right.
It's just arguing that the firing fields repeat at some frequency in different directions.
Yeah.
And
so at the same rate.
Right.
Just say that parse that sentence again.
It's just arguing that the firing fields, right, as you know them in the 2D lattice, they repeat.
Yeah.
Right, with a certain frequency in different directions.
Oh, this is the frequency in space, not the frequency of the cell firing. Okay, well, right. Don't. Okay, you have to, you got to bring the audience along.
I'm sorry.
Moving
too fast.
Just be patient because I'm slow.
Okay, good. All right, so this slide is just simply putting these, like 2D and 1D, sort of contrasting them with one another to familiarize you with the idea of, okay, we turn spikes into firing fields. We can look at how these firing fields overlap or don't overlap, and we can do a Fourier analysis to take a look at what are the periods, right, with which we can understand these firing fields to repeat. And so one thing that is, for example, rather obvious from the fact that you have multiple spatial peaks here is that you will not get very regular oscillatory periodic spiking because there are multiple frequencies in here. So the firing fields are not generated by one, standing sine wave.
So let me see if I understand this. A single cell, if I just had one frequency peak in the lower right-hand corner, I would see equally spaced.
Right.
But the fact that I got multiple ones, I'm going to have this sort of, discordant.
Right, it's more complicated, right?
Yeah.
If you had one peak in the 2-dimensional environment, then you would see very nice stripes, right? Yeah. But you don't.
So why do I see very nice
generated
in the 2D environment? Wouldn't I have the same problem there? That I have different frequencies that which an individual cell is firing at different spatial frequencies?
Yes, you do.
But we don't see that now.
Yes, but yes, we do. The fact that there is a 2D lattice, right, is the result of the fact that there are 3 distinct spatial frequency components to this. The grid repeats in 3 different directions. There's 3 vectors in this field, 1, 2, and 3.
So now you're saying those different frequency analyses are actually oriented in different directions.
Yes.
Well, it's not obvious. I could have all this because you're saying in the bottom one, in the 1 dimension, I'm going to see the same 3 frequencies in 1 dimension. Therefore, it's going to be kind of irregular looking.
Well, that's the difference with in 2D, you can point the oscillation into a direction, right? Whereas in 1D, you only have the 1-dimensional expression of it.
I got it. I'm just trying to understand it.
Good.
Come on.
Good.
Bring us along.
Yes.
Okay.
All right. Moving on from the first figure. Let's see. So, if you want to understand, so this is completely theoretical. This is not real data now, right? This is theoretical idea. If you had a perfect 2D lattice like this, and you decided to walk through it at different angles. So you have this data, and you're going to go through it. What would be the firing rate that you would expect?
Yeah.
So if you go at the 0-degree angle, then if you only walk along the direction of one of the spatial components of this grid, right, then obviously you get a very nice regular oscillation of the firing rate. That corresponds to one big merge peak here, right? That is the main component. If, however, you start now drifting this gradient, right, then the expected firing rate along that path starts to change, right? And you get all kinds of peaks of different amplitude, and it becomes very obvious that there's now diverging. So you see how this original peak F1, F2 actually splits into two, because you now also get the component in this direction. And so now you see how by varying the angle and the power spectral density, you see these peaks merging, right, from 2 into 3 into 2 into 1 again. Once you arrive at 30 degrees, you again get a regular oscillation at a higher frequency though, right? But it just goes to show that you can do this kind of analysis, and you can somehow, if this was ideal, you'd be able to infer the angle at which you are going through the lattice from a firing rate plot like this. This is very nice and clean because it's all theoretical.
Could I make one quick point?
Yes.
That sampling that you're doing in this perfect lattice also corresponds to the problem in computer graphics of aliasing. So you actually get some of these components wrapping around and appearing as low frequencies because of that. So it's even more complex than what it appears there because of that wraparound effect.
Yeah, the problem is, of course, these tracks have a limited length. So you, like, theoretically, to infer all these frequencies perfectly, you would need an infinitely long track. In practice, you don't.
So they wrap around.
It's very rarely do animals actually pass through multiple fields, like so many fields like this. It's like
exactly right.
People didn't see grid cells because the grid cells, the bumps are so far spaced apart that in most environments you don't get a repeat.
Yeah, we saw that here, right, where there's a limited number of fields, and even in the 1D track, we see like 1, 2, 3, 4 bumps here on the 6-meter track. So that makes it very difficult to infer that there are, in fact, 3 frequency components to this. So now the interesting idea is that if you do parallel slices through a grid, which is going to be important for understanding co-modulated cells, I'll talk about that in a bit, then you would expect that while they have a different phase, depending on where they start, they have the same power spectral density because they traverse it at the same lattice angle here, right?
As long as you go through enough bumps.
So exactly. So that means from the power spectral density, right, from the spatial frequency analysis, right, of the 1D plot in an ideal world where such 1D slicing would work, you would be able to infer the angle by which you are traversing the lattice.
Okay, so this is an explanation for the first figure.
So that is one perspective one might take, and it's in fact the hypothesis that they are testing.
How's an explanation?
So the first figure, you had 2 electrodes that were being measured, that were being reported from. And they say, well, suppose it's really a 2D grid up. It's really a 2D module.
Yeah.
You have 2 electrodes, they have 2 slightly different phases, and the animal is going in 1D along something. Then you would see the spatial frequencies of both of those lining up exactly, even though their actual firing would be all over the place.
I see. Okay.
Their spatial frequency.
Basically, the actual firing doesn't line up, but
the spatial frequency.
Got it.
Which is exactly what you would expect if it was 2 nearby cells going in the same direction.
Got it.
It doesn't mean that's what's happening, but there's a possible explanation.
Exactly. And so you can, in fact, very nicely, so this diagram kind of puts all of these different metrics that we're going to see now in different arrangements kind of neatly together. So you have the 2D lattice and a linear slice through it. So from that you can infer, if it's true that the firing rate is generated by that slice, then you can infer the firing rate because you're passing through these firing fields here, and every time you pass it, your firing rate goes up. In both of these cases, in 2D and in 1D, you can do a Fourier analysis. So in the 2D case, that means you get these neat little bumps. in the 1D case, you will get 3 bumps, because essentially what you're doing, mathematically speaking, you're projecting these firing fields onto the line. You can think of it this way. You're gonna, by moving at this angle, you're gonna pass by, right, these different Fourier fields. And so when you're very close to them, you get a big peak, right? And when you're further away, it's gonna be softer, right? But in some sense, you can, like, all of these are connected, right? So you could infer the 1D power spectral density from the 2D power spectral density and the angle, or you can infer from the firing rate the angle in the 2D lattice. And so, like, all of these are related, right? Under the hypothesis that the 1D response is explained by a 2D lattice slice. Right. So I guess what you see here in these lower 2 is exactly that idea of passing at the same angle, and then you get the same frequency components, but depending on where they start, they might look a little bit different. I think we're going to see this better in further ways. So now, an interesting way to sort of look at the data in a slightly different way is to, instead of looking at the frequencies, which are hard to determine because these tracks are not long enough, you look at something that's a lot more easy to measure, which is the gaps, right? The gaps between the firing fields. And of course, we know that if you move along the lattice, right, in an ideal world, right, you have the sort of the spatial factor A, that is the distance between your firing fields. But if you move at some different angle, then you expect a lower frequency, or put differently, you expect a larger gap, right? So if you moved at this angle here along that line B, you would see a firing field every square root of 3A instead of every A. And so if you now take different lattice slices through here, you in fact expect very particular gaps. And the cool thing is, of course, since all of them are scaled by A, you can in fact describe the whole thing by the gap ratios, because those are then invariant to the field size. And so by looking at the gap distribution, you might also be able to infer the slice parameters.
Hey, Florian, can I ask a question? Can you hear me okay?
Yes, I can hear you. Wonderful.
So would it be true to say that, this is maybe a dumb question, but would it be true to say that then the grid in this is just an artifact of then oscillatory dynamics, or just oscillations then? So it could be encoded in oscillations of a cell?
Well, of course, you can ask the question, how does the field get, like, could you generate that field through those oscillatory components? That is sort of like, I mean, it's...
Yeah, that's what I'm asking.
It goes either way, right? I mean...
Yeah, because this seems to go the other way. I always think of it the other way. This is sort of taking it in the opposite direction.
Marcus is shaking his head.
I don't think there's any mechanism here that's been suggested. Like, if grid cells have firing fields like this, here is the result you're going to see. That maybe it's happening because of oscillations underneath, but like you can't conclude anything about that from this. This is just what's going to happen no matter what the grid cell model is.
Right. So this is sort of a theoretical idea of, like, to make it a little bit more tractable. And so now you can look at actual recordings. So 1D spatial tuning curves. So these are just recordings, right, from rodents in 1D tracks. And as expected, you do see sort of like these firing fields. So they're often firing at the same position in the multiple different labs that you see in horizontal lines here. There's in fact 2 different databases here that they're passing. So like one of them has like a 6-meter track, the other one has a 4-meter track. And you can take a look at the firing rate, right, from that across the labs, and so far as there are, as there's no drift or no sudden change, right? Those are going to be very nice.
Are the first 2 from the same animal, just different electrodes? First 2 columns.
No, what makes you think that?
They're both 4-meter tracks. So
yeah, I think it's just the same data set.
Okay, but different animals.
Yeah, I think there's some number here. I don't know exactly how to read this number. I guess the T is a tetrode and the C is a channel or something. and then maybe this is the animal number or the trial number. I'm not sure.
I'm just wondering if they know if it's 2 from the same module or as the first step.
Right. So using a bit of math that we don't need to go into much depth. I hope I have convinced you that somehow from this analysis of the firing rate and then looking at the power spectral density, we can infer certain parameters, like for example, the angle at which these different firing fields, right, would be explained by the idea of going through a 2D lattice.
I might be able to interject something useful here to make it seem even more worthwhile to understand this. You'll see where I'm going.
I like that.
The later Inferring paper, the one that talked about like how grid cells are distributed in the cortical sheath, that experiment happened using rats running in 1D. They used this method to go back and reconstruct what the phases of each recorded
Is that right?
grid cell
This is
must
the one
be.
that I like that had the picture. That was all
There
done
was
one-
no 2D experiment there. That was all done using this method to infer
Wow.
what the phases of the grid cells were.
So they assumed it's really 2D.
Yeah,
You're seeing
they assumed
projections.
that they kind of, in this paper, they kind of proved this technique. And then they used the technique to go and figure out like, the distribution of the phases in the cortical sheath.
Yeah, we actually get to determining the phase 2 in a little bit.
Okay, yeah. I was throwing this in here just to say that this isn't just a method that was isolated to this paper that you could forget about after 20 minutes from now. Like it's something that is.
Now I'm struggling to make sure, because I might have maybe misinterpreted that later paper. I don't know. I mean, I didn't know that item that you just mentioned that they were measured in 1D. So that's another level of sort of abstraction in some sense to understand that data, which I didn't know. Thank you for bringing it up. But now I'm beginning to question whether I understood it prior. No, it's good. I need to know that, right? Ignorance has to be corrected. Interesting. Okay.
Right. So to just briefly step back a little bit, one of the things, since this lattice gives you like these characteristic lengths of a square root of 3a and square root of 7a, what you are expecting in terms of gap ratios are 1 over square root of 3, 1 over square root of 7, and square root of 3 over 7 as gap ratios.
Yeah.
So if you now do the analysis on the gaps between the firing fields in these plots, you are expecting in an ideal world to find very nice peaks around these ratios of 0.34, 0.59, and 0.58, right? Which then would correspond to these different peaks. This, of course, does not tell you yet whether there's a correspondence between the 2D response and the 1D response. This just goes to show the idea that you can somehow fit, right? I think that's the green line is the fit and the black is the real data. You can somehow fit the firing fields, the firing rate of the 1D response quite nicely with an idea of just finding a slice through a 2D grid that would yield similar firing fields in similar positions and with similar amplitudes, right? Containing the same frequency components and also somehow fitting the gaps between the firing fields. And so that, of course, you have to control whether that is just maybe that's just a very powerful thing to do, and any firing rate distribution on a 6 meter track can be fitted by a slice if you are free to choose your face and your angle, and who knows. So they did some controls by creating artificial data that were either gap randomized or like completely randomized, so where you just move the firing fields to random locations to preserve the number of firing fields, or you shuffle the gaps. And they showed in mathematical terms that the fit is not nearly as good for those. And in fact, it's quite a bit of a difference. We don't need to go into this exact fit of quality metric that they have. They have a score that they call power spectral density peakiness, this P3 number, which somehow assesses whether there are 3 strong peaks as predicted by such theory. But the worthwhile thing to look at is, for example, this complete aggregate of data that we have in D here. Let me just quickly see the rate. So that's the aggregate gap distribution pooled across all cells in the data set. So this is like a massive average after the gaps in each cell's response are normalized by the lattice period, right? So you take out the lattice period. So that means that the lattice is 1 now.
Yeah, it's a ratio.
And so what you're expecting is big peaks at square root of 3 and square root of 7. It turns out the peaks are a little bit earlier than that. But there are 3 nice peaks. So it's, not out of the world. And judging by the statistical analysis that they have, this explanation is way more likely than not the right one. Now, all of this just shows that we can fit the 1D responses with this method. It does not show us there is a strict correspondence between the 2D and the 1D response. One way to assess that is to look for co-modulated cells, so in the 2D grid, and see whether they behave accordingly in the 1D grid. So you have 2 cells that are part of the same module and that have sort of the same firing fields. So for example,
You're saying recording the same 2 cells in 2D environment and 1D environment?
Yeah, exactly.
Okay.
So here you have 2 cells, first in the 2D environment up here. And then, no, that's actually not it. Let me see whether I get this right. So yeah, so the idea would be that cells that are co-modular, so that's like fire together statistically, right, should ideally be parallel slices with a similar scale factor. So because their angle would be the same, so they would have the same power spectral density. And you find some cells for this holds true, and then you would infer the angle from it. And the question is is it true that all co-modulated cells then have the same slice angle when you try to extract the slice angle like they did here, right, from a 1D linear
I'm not sure what
response?
what am I gaining from this? What, by definition, I expect this?
It's a proof of it's one more way to prove that this is a correct way of understanding, because the prediction is if you can infer the firing rate in the 1D case from the 2D lattice by slice, then cells that are co-modulated in the 2D case should be characterized by the same slice angle in the 1D fit.
Not to diminish this, but that seems sort of an obvious thing, right?
Right. So it's just needing it to test.
Just doing it, and it's like just doing the same thing.
Because it sounds like the previous data were not necessarily from the same from 2 cells in the same module.
Right.
Here now they actually have 2
But
cells.
we would expect this under all the explanations of grid cells I've ever heard, would expect this to be right.
Right.
Okay. So it's sort of a baseline verification again. It's not like presenting something new.
Right. Okay. So you see 2 pairs here, one up here in A and B, and one in C and D. And so these two cell pairs have very similar power spectral density, as do these 2, very similar power spectral density. And so the model would suggest that, in fact, if you infer what that means as a 2D slice, that these are parallel slices. So they have a different phase, but they do have the same slice angle. And so if you now look at the distribution in the recording, what are the slice angles that you get out and what are the scale factors that you get out, you get a very nice distribution. The problem is that in this uncurated data, you don't always get the same slice angle. So you have 2 co-modulated cells that apparently are not explained by the same slice angle. They sort of fix that with a correction where they say, look, if we restrict our analysis to looking only at, what do they call it? See.
Just like the cosmological constant.
Yeah, something like that. So they find an experimental procedure to pick sort of a consistent set of peaks in the power spectral density and infer the slice angle only from those. And then it turns out the fit is quite a bit better. To me, that looks a little bit like cheating. I also find it problematic that there's sort of a gap here at 0 to 10 degrees. There's very few cell pairs in there. But other than that, it's nicely distributed.
Oh, right down the bottom.
Right, so there's not a lot of cells that would be predicted to have a 0 to 10 degree slice angle. So for some reason in the data set, they don't have a lot of cells that are going along the exact lattice orientation. So the firing fields as a consequence, are not very periodic. But in fact,
there's an extra question here is how do the grid cells get aligned with the object that you're modeling, right? They get anchored. But without knowing what that anchoring is, one way you can anchor is say, I'm always going to anchor one of those on that straight, most repetitive, or you can say, no, I'm going to anchor opposite. So not knowing what the mechanism for anchoring is, then that gap could be easily explained.
Right. So anchoring matters. And so I don't know if they did this sort of like early on or whether reviewers poked them at exactly this question. You actually have a plot here of the grid orientation versus the slice angle that is predicted for these co-modulated cells. Again, the cells that are co-modulated are in the same color here. So you would expect them to have the same slice angle, and you would expect them to predict the same grid orientation. And so this seems to work quite well.
So that means that 0 to 10 degree gap may not be a problem.
It may not be a problem. Also, keep in mind, these are very low numbers, right? These are a couple of cell pairs. I think they had 150 recorded units. And then, of course, you choose for the ones that are actually qualify as grid cells, and like the numbers go slower and slower,
In fact,
lower and lower.
It's almost certain that the anchoring of if I have 2D grid cell module, the anchoring on a 1D space is not going to be random. It's going to consistently have some sort of consistent base, because it's based on sensory input. So it's not going to be random. It seems unlikely. It seems like it would be some bias, and therefore you'd have a bias in that angle.
And you wouldn't want the modules to anchor along the grid, because you would waste a lot of your grid cells. They'd never be reached as you're walking along this track.
That is an interesting theoretical argument, yes. For like making good use of your cells, yeah.
But in all cases, I mean, if I'm in a 2D environment, I still need multiple grid cell modules because I can't really tell where I am. And so if I had multiple grid cell modules, then I could compensate for the fact that the 1D module is some of the cells are being wasted. I'm just saying you would still know what you still have enough information to resolve it. So it's a good argument, Marcus, but it's not like a requirement.
I'm a little confused. Haven't we seen papers where the animal runs along a 1D track and you see very periodic responses?
I thought so too, but
that they seem to say you don't get that.
Well, at least I mean, this is not like big enough in numbers to really say much about that in a statistical sense, right? I was just struck that somehow.
I think some of the plots you have in mind right now were theoretical, where they showed that like under this model, this is what you would expect in 1D tracks. I think there might have been some early ones that had some suggestions of that, but really in the past decade, they've been more like this, or even more messy than this.
Okay, I kind of remember too. I kind of remember them saying, "Oh, we measured it, and this is what it looked like."
Including like these Y tracks.
Yeah.
And,
they had ones. Yeah, that's what I thought too. But maybe you're right. I don't know. I guess I wasn't looking at it that carefully. But I had this impression that it was measured nice periodic responses.
it could also be that as an experimentalist, you keep poking at cells until you find something that's periodic.
I don't
and ignore these
There
missing
might
ones.
be biases of all kinds in these data sets, right? They're curated in some sense, so it's very hard to talk about this in raw data analysis terms. Anyways, moving on a little bit, one other cool thing that one can do. Why is it jumping like this? Why can't I just? Okay, let me see. Now it will let me scroll or not.
This also doesn't say there aren't 1D grid cells.
know. No,
Right.
It just
No, no,
says that there are.
Can we-
likely that the 2D grid cells in a 1D environment look like
Can we
this.
move that discussion to later when we're through? It's
Okay.
only 2 figures left.
Okay.
Because of course that's a big discussion.
Because
to be
I
had.
think something that came up after the conversation last Monday was, "Come you have be patient." Something, this is a discussion more than a lecture. So
Right. That's okay.
that came up after when I first described it. I said these modules could be equally distributed in space, and then later on we had a conversation. Maybe it was just you and I, though. So they could be aligned. All the 1D modules could be aligned in 60-degree angles, and that might explain it. Which I think would still be consistent with this.
Right. Because we have a lot in the hypercolumn, and so if you only need 3 angles to span the space, right, for the 2D lattice, that means you have many in each 1D dimension.
Yeah. Okay, so that's where I want to go. I hope you're going there.
So one more thing one can of course do that we didn't talk much about yet is the relationship of relative phase predicted from 1D and measured in 2D. So you can infer, right, from the 1D using the mathematical analysis of the 1D firing rate response. So you take a look at this is too bad. I can't really do this well. But anyways, you've seen the 1D firing rate, how it like evolves, right? And so you can infer not just the angle based on the power spectral density, or the gap distribution for that matter, but you can also make an attempt at inferring the phase from the 1D. And the question is, does the 1D predicted phase actually coincide with the phase in the 2D response?
It should.
It should,
So
if
this
this
is
theory
another
is true.
baseline test, right?
Right, it's
If their
another,
theory is correct.
Yeah.
if the theory is correct.
Yeah.
And so because of what's called, because of the degeneracy that is inherent in solving this, when you solve these equations, you actually get 12 solutions back for the phase. And so you kind of have to pick one. But if you pick the one that fits best, then you get this very nice pairing here between, let's see, what is it? The relative phase. So this is now plotted right in the rhomboids. So the phase to 0.5 to minus 0.5 spanned, predicted in colored triangles. And then the actual value that they see in the 2D data is the circle. So you would hope that all the triangles and circles of the same colored cells are always very close by, right? And of course, like you can see that, okay, that works reasonably well. I see a lot of pairings here, right? If I go through this diagram, but of course you have to do a statistical analysis, whether the prediction error magnitude density analysis against controls and whatnot. And so they show that this is more likely than not the correct explanation. Didn't quite understand the mutual information thing because I didn't have time to, read into that. But apparently, you can do an analysis on that. And then I think the last thing that they did. Yes.
No,
Yep.
Don't go back up again. I'm going to read the text. Right,
The text.
right there. Stop there.
Right.
I'm reading that shifting one. Do you want me to read that?
All right.
I don't know where he's reading. I know I'm muted. I'm only saying this to the video audience.
Mm-hmm.
This research meeting was filmed before a live studio audience.
I will show you. If you let me present, I will show you.
Read it first. I'm sorry. I have to.
These are live raw interactions between our researchers and Jeff Hawkins, our founder, who's, I guess, the chief researcher.
Okay. Mm-hmm. So what you sometimes see in these recordings is this, where a firing field seems to drift over time. So these are laps, right? Same electrode, same animal, same tetrode recording, right? And what you see is that over multiple runs, this firing field, in this case, drifts
Can you
backwards.
explain the experimental setup here? Is this like a 1D track? The animal is going around and around in it? What does that mean?
Yeah, I don't know. I would have to take a look which exact data set this came from, but they had a virtual track ones, which you can actually reset. So if this is the virtual track one, then it's very easy. I mean, you just let the animal run to 6 meters and then push a button and you reset it, right? And
So it's
another
just-
chance.
Basically, it's going down a long corridor where the patterns change at this period of time of the length of this.
Mm-hmm.
And so it just starts over and over again.
Right.
Um,
Multiple trials.
okay.
And what they see sometimes, not always, but what sometimes happens, which you can now analyze with this new lens, is these drifting firing fields, where they seem to drift at some moderate rate. Because it seems it's not just that the first one here is sort of drifting backward, but actually the second one is drifting too. And the third one, the fourth one, it gets a little bit messy to analyze this without data analysis. But you see this actually in several cells, right? So this is a different tetrode, and it also seems to be drifting
Interesting.
backward.
It looks, if I were just looking at this data, I would say by the time you get to the end of the track, the drift has changed. It's back to normal.
Right.
Those two vertical ones at the end. It's almost like if I could think like a rat, I'd be going, oh yeah, I know this place. I'm anticipating what's going to happen. I'm sort of one step ahead. But when I get to the end of the track, that's the end. I know I'm at the end. Right. There's sort of a mental aspect to this. You can imagine yourself feeling that. It's like I'm always racing ahead and interrupting you while you're speaking. Because if I think I know what you're going to say, I just
That's okay.
If I didn't know what you're going to say, I'd have to listen. I have to parse it as they come in. But if I think I know what you're going to say, then my brain goes ahead of it.
Oh,
That
right,
could
like
be
a
what's happening.
procession of sorts.
Well,
Yeah.
this is just the rat saying, "Oh, I recognize this place. Yeah, 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 yeah. Let's get to the end. Oh, not the end." So it's interesting though that the very last bar is vertical, not tilted.
Yeah,
And
I
most-
don't know if it's true that it's in general it's a phase procession of sorts. That
I would not even try to use that term, but
Okay.
maybe. I'm just pointing out that the biggest slope seems to be at the beginning, and at the end there doesn't seem to be a slope.
Right. I don't know if that's generally true. I mean, you could answer this with a lot of data, I guess.
Well,
It's an interesting
I'm
hypothesis.
just looking at it right there. That's what it looks like on those
Yeah.
two.
Those two cells look like that.
Yeah.
Yeah.
And anyway, so I could even explain, in some sense, how that occurs. It has really nothing to do with the actual mechanism of the grid cells. It has more to do with learning that these things fire a little bit earlier.
Right. So when you do now the lap by lap analysis of the inferred angle, the inferred scale factor, in the inferred phase, which obviously has two components, x, y, from the 1D data, right, trial by trial, then you see in fact that all these different trials predict the same angle. There's a little bit of a blip here up and down, but sort of all the data suggests that this is going at a 20-degree angle through the grid. And they predict, pretty much a constant scale factor too. The one thing that seems to drift slowly but steadily, and interestingly enough also linearly over the lap numbers, is the phase, right? And in fact, these two cells drift in parallel.
Is that the same thing we were talking about above there?
Yes. So if you now do the analysis, right, on lap by lap, and you try to infer the 2D angle, scale factor, and the phase, then in fact, this method of analysis explains this data as saying, look, the only thing that is happening here is that the angle and the scale factor stay constant, but the phase is shifting. So the right way to view this is this figure here, where essentially what is happening is that this is the power spectral density here, right, and the gap distribution and whatnot, but this kind of shows that there's a drift over time. And of course, it's an interesting question whether it's true that drift tends to be negative in general, or whether that's just true for these particular cells. I don't know that, but I guess
I guess what I'm saying is,
right.
I give an alternate explanation of the same data, which sounds less highfalutin than the phase is shifting over time.
Right. I mean, this is just the mathematical analysis.
I know. I need to say these things because otherwise, we just run right by, and it's like, be careful.
Right. Apparently, what also sometimes happens, particularly on tracks where there are landmarks, is that these things can change rather abruptly. Because apparently, the re-anchoring can happen and whatnot. And so they nicely chose data sets where there are no landmarks, so there's no remapping happening, where they chose the cells that are reasonably consistent. So there's all kinds of pre-filtering of the data set in some sense. But what they do see a lot is apparent drift. And the interesting thing is that drift doesn't change the angle or the scale factor. I mean, we'd be surprised if the scale factor changed, because that would mean you would switch the module that you're working in. That doesn't seem to happen.
So this is one way. Another way to look at it, you could test this, because the hypothesis I threw out is that this is just because the rat is learning this environment, and the more it learns it, the more it anticipates what's going to happen next. But so at some point, after some number of trials, you assume that there'd be less learning going on, in which case it might stabilize again.
It could also be there's no landmarks. There could just be error in the sensory motor prediction.
But why would it be shifting over multiple trials?
It's just a constant. You're just constantly making mistakes. I mean, mistakes grow over time. And so.
Well, no, but it is anchored at the back. And that was that one. When you get to the end, you're at the end.
Oh, it's not an infinite track.
No, I don't think so.
Okay.
That's why I was proposing at the very end is vertical. The rat goes, oh, I'm at the end. I know where I am, but it's like. Imagine you're going down the hallway at night, going to your bathroom, and the first time you go down, you're going really slowly, like, where am I? And the next time you go, I can imagine where I'm going to get. When I get to the end of the air type of thing, right? You're sort of anticipating anything. Anyway, you could.
That's a good hypothesis, and experimentalists should be able to test that.
Yeah.
I don't know. I mean, like, ideally, would have David Tank on the live stream. Actually, wrote an email to Eli and Tank and what's his name? Yoon, the first author on this, So I don't know if they, will get back to us, but it's an interesting question. Yeah, and that was pretty much it. And so it turns out it's reasonably well explained by 2D slices in terms of statistical analysis. It does seem to hold true. And it's a productive way of thinking of 1D responses, is thinking of them in 2D slices when you're recording entorhinal cortex in rodents behaving.
I guess the question I would ask is, you're saying, okay, if it was 2D, you could explain it. You could explain, through 1D slices. Is it? The question I would ask is, are there any explanations where it could really be 1D grid cells and you would still see this kind of data?
Right.
Are there any versions of 1D cells?
Why wouldn't I have a different question. I don't understand why all versions of 1D grid cells you would show this if they were at 60-degree angles. So if I just took 3 grid cells, if I just said these are 3 different grid cells, modules. The modules, in the module I'm saying I got a little 1D module. And so there's a bunch of cells in that 1D module. And let's say that I have 3 of those modules and they're oriented at 60 degrees from one another, right? Now, that module is only thinking in 1D, but I think it would still exhibit all the same things. Why not? You said no.
Any kind of 1D module, each cell's firing field is going to be this big sheet that's at some orientation in space.
Yeah.
Any straight path is going to be periodic sheets. Any straight path is going to give you periodic fields.
Yeah, it would be strictly. It would be. You're saying it would be strictly periodic.
Yeah.
Is there anything where they tried to, I mean, if the rat is running down a linear track, is there how would it pick the base angle? I mean, wouldn't there be only small deviations from whatever that base angle is, orientation or whatever? I'm just trying to figure out which of these angles in a linear environment are even accessible.
All angles apparent, like at least according to their data, right? There's a very broad distribution of the angles that they get from the 1D response. If you look
the
at
first few are not there.
Except that I think for some reason there's a gap here in the first, 5-10 degrees. But other than that, this looks very nicely distributed.
I think
the question I have is why? I mean, if the rat is running down something, why does it need all these angles?
I don't know if there's any theories that are coherent. Maybe Marcus knows them, or you, Florian, about how these get anchored. We know that they're anchored, but how exactly is the question where I think you're asking. Well, how do they get anchored this way? I don't know if they know that. Right. Are there any working theories for them?
I just vague ideas. I'm actually a little surprised that it's this well distributed. Usually the orientations are actually like a few clusters. Usually it's not uniformly distributed. So this is more than I expected.
There was this other question. So if you look at that the tank paper that we've talked about in the past, where they show, all right, there was these phase clusters, right? So in a module, you had like 6 phase clusters. So those 6 little grid cell modules within a module. And they show that the activity of those individual phase clusters was not explained by position. That is, individual bumps were, if I looked at the 6 phase clusters, some were active and some were not active. Which was that was sort of an additional coding on top of all that. Did that, I mean, given some of the same people, did that come up at all here with that mention?
I'll just throw one thing in. I did notice in the text in that talking about bumps being missed or
that's what,
I did see in the text here just briefly as you scroll by that I was interested to see that they talked about it all over here.
Yes, yeah, that's true, and I do talk about that.
So that I thought was, as I've said before, I think it's a huge clue, right? What do you say? Oh, misfield. Yeah, is that the term they use? Misfields will reduce the height of the gap distribution.
In the power spectral density.
So they're saying it doesn't matter if there's misfields.
Yeah, but I'm saying the misfields, what they showed in that other paper is the misfields, I think, were consistent. It wasn't like some random lack of firing. It was like an encoding of some sort that was. So they're basically saying we're ignoring that here. Even we can ignore it and we still get the same results, which I believe.
Yeah.
But that doesn't mean that we should ignore it.
Yeah, this is the one paragraph where they talk about missed fields. One thing, of course, that I was thinking about, like you mentioned this idea, is there any way that you would get the appearance of a 2D lattice, even if fundamentally, like units close by are actually 1D? Is that possible? And I think the answer to that is to look at patch clamp recordings of grid cells, because that way you would get you would know that this is not like coincident by the electrode, but it's actually the cell itself. So so that's why I was looking at the. I already got like one of the underlying data set. This is not the Constantinescu one, but the other one. This was one of the raw data sets, the membrane potential dynamics of grid cells. So I just started reading this one.
Yeah, so this is the idea. Maybe they're really recording multiple cells when you can recording one cell.
Yeah, exactly.
Yeah, I wouldn't think that's the problem, but I don't know.
But I don't know. I can't answer yet. I just started.
I wouldn't think that's the issue.
That is actually one of the data sets that they use for this.
They're usually pretty good at determining this. They're usually pretty good techniques for knowing what cells they're recording from.
Yeah.
And so that local effect doesn't usually fool them. So I'd be surprised if that was the case.
Plus, a lot of the recent data is also with calcium imaging, and with that, you actually see the neurons.
Yeah. Again, what I find is I'm trying to reconcile these fundamental problems that we have. If I think about encoding location, there doesn't seem to be enough information in a patch of entorrhinocortex to represent location. There's, the grid cell module, the set of phase clusters is fairly large. And we all know that one grid cell module is not going to be sufficient. And even 2 or 3, given how noisy they are, apparently noisy they are, and so on, it's not sufficient. You really want a fairly large population of grid cell modules to choose from to represent space. And it doesn't seem to be there. And again, when we look at a patch of neocortex or something less than a square millimeter, it's also hard to imagine how you're going to have grid cell modules in there. So and then we have I don't have any good idea. I can't say no one, but I had trouble really figuring out how you take like an orientation, a 3D orientation, how do you represent that, and then how do you take that representation to know how to update all the different grid cell modules correctly. That's a challenge too. And so when I went down to 1D, I could solve all those problems. Right. And yet, on the other hand, that doesn't what I agreed when I said last Monday. I said it doesn't look like this. That's not what we see. We don't see 1D modules. We see 2D modules. And so I'm still trying to reconcile these two. I'm still trying to find there's a way of taking the ideas I had last Monday, which were like, hey, I can explain how I can represent end orientations, end dimensional orientations, end dimensional grid cells, and figure out how they should all update appropriately as we move. I now have a neural mechanism for all that, but it requires this 1D structure, which I don't really have any evidence for.
Keep in mind, this is all entorrhinocortex and all behavioral rodents.
So so we had, and then you and I discussed. I think there was several possibilities. There was like one possibility. Okay, in the entorrhinocortex, they really are 2D, and there's really some complex mechanism there. And maybe the exact same thing's going on in the cortex. So they're both like that. And we just don't understand it yet because we don't really understand how that works. The other possibility is the entorrhinocortex is not the same as cortex. It may be cortex, which is really, I've never heard any evidence that entorrhinocortex is created with sort of a mini column ontogenesis idea. Maybe it is. But we know that the cortex is built up with all these mini columns in ontogenesis. And therefore, maybe the cortex works on a slightly different structure. It's not the same as entorrhinocortex. Right. But then now we've seen like fMRI data that suggests that there is a 60-degree orientation in at least some parts of prefrontal cortex, although it's weaker than in the entorrhinocortex. So that was the Constantinescu thing. Or the other possibility is that maybe both of these systems are really one-dimensional. They just look two-dimensional for some reason that we haven't figured out yet. But there is, we can't be complacent with the 2D module idea yet because we just don't, it's not an explanation for how the whole system works. We just don't have the pieces.
No, none of this makes a functional argument how this works, right? This is all descriptive, and then in this case, putting a very nice mathematical model on it, which turns out to work well, right? which I found very nice. I mean, this paper was a blast to read just because it's so nicely graphical, because, it's like plot these lines through the 2D lattices and try to understand what is happening.
Yeah.
So there's an amount of topological, intuitive sort of understanding here, right? But there's many different ways to pass these, and that doesn't say anything about functionally how this is set up.
So let's talk about the missed cells again, if we have time. I don't want to go beyond our prescribed time here. What are we up to? What time is it?
11:40.
Oh, we already went past on time. We can take a top line if you want.
Right.
I think we stop there. But then for those that want to continue talking, we can talk.
Okay.
Yeah, I'm gonna
Cool.
head off to-
Right, sorry about that.
And, you guys can continue.
I guess so.
I'd appreciate it.
The mist, whatever field thing, is it going to be? We have no idea what's going on there. We can brainstorm about it.
That's what I'm thinking.
That's what it is.
That's what I'm
Okay.
thinking.
Okay.
Right. Like, because I saw that it didn't seem to be random, but I recall from the tank paper that the mist firings were sort of consistent. And so it was like, okay, that cries out for an explanation of some sort.
And that one I'll throw in. That's been replicated in a couple different studies, including ones where rats are moving into
Oh, is that right?
where some of the fields are like almost not present, and other ones are.
Some folks had to take off,
Actually,
but they let me keep live streaming.
more information about the rat's location from looking at a single module than you would expect.
Okay, so I learned one.
Sure.
Right.
So I'm not aware of those other papers, so I would like to see them. The only time I've ever seen that was the tank paper, which is about a year ago, I think, right?
Yeah, this
So you
one's from earlier, from Kate Jeffrey.
Earlier.
Marcus to move.
Yeah. Well, this one about the local, about being able to decode local information.
By the
fact that
missed
these-
cells?
Yeah.
Because when I talked to Tank, it came across as that was a new discovery. But you're saying it's not. It was
No, in some ways I would like I like the other study more because it's with rats moving in 2D, whereas the tank result is virtual environment,
Yeah.
virtual tracks.
Well-
Just to quickly point out how this would look like in 1D, right? This cell, right? The grid one would be the prediction from the 2D slice. And it turns out there's no bump here, even though the prediction says there should be one. And this bump is half the amplitude that would be expected,
Yeah.
right? So maybe this is what we're talking about.
It might be, although I think the one paper I'm familiar with, it was consistent. It wasn't, yes, it was like that. Yes, it was varying levels. The missed ones, if I recall, maybe you remember, Marcus, what I recall it was a graded missing. Sometimes they were completely missing, sometimes it was half as much, but it was consistent. And that was
I see. That is a bit of a.
Well, that was the thing, because now we had the possibility that a single grid cell module, or maybe just a couple of them, which is what seems to be you only have the room for in the amount of entorhinal cortex sheet, that now you have this additional coding scheme on top of it, which is Marcus was saying. What I was saying, that's an additional coding scheme. Marcus said that there were previous papers that can tell you more about where the animal is. So I thought that was, I'd like to read that because I think in the tank paper I read, there was no speculation about or even talk about what it would be called. It was just like, yeah, they missed, but that was an interesting observation. But they didn't do any analysis of it. They did some analysis, but not that kind of analysis. So that's an interesting idea. I feel that's like an important, it cries out for explanation perhaps, as maybe a way of resolving these differences. One thing I speculated, which doesn't, I see your point, Marcus, this data doesn't fit with the 1D hypothesis, but the idea that if we did have a bunch of 1D modules, then you could do a sort of a sparse sampling or coding from them to infer state of the object, which is something else we have to do. We have to say we're in a reference frame with some object and we're at some location in that object, but we also have to infer the state of that object, like the cell phone state or the stapler state or something like that. So I was excited about that. So that's another sort of complication going on to this. But I would, if you could send me one of those earlier papers that you thought looked better, I'd like to look at that. Who did you say it was again?
Kate Jeffrey.
Kate Jeffrey.
I don't remember the first author, but I can send it.
I appreciate that.
And so okay, a couple thoughts just came up. One is you're talking like certain problems just vanish once you use 1D modules, like problems involving orientation. And I think I don't think any problems vanish except for the number of cells you need.
Well, in my mind, I can now understand an answer. So, for example, just how would here's where I don't know if you heard what I did last week, but
A little bit.
okay. So if you have a 3D, let's say you have a 3D orientation in a 3D space, right? I don't know if that 3D space is represented by a bunch of 2D grid cell modules at different, like how we represent that, maybe with different slices of 2D that we're representing that space by. But then anyway, I have some sort of 3D orientation. And of course, that 3D orientation dictates when I move, how each of the grid cells module update, right? Very precisely. They sort of, different orientations, the bump moves in different directions, and I have to make the differentiation between the different grid cell modules. So I have this sort of, mathematically, it might be easy to explain, oh, I have a 3D orientation, I have a 3D space. How should my bump move in 3D space based on the orientation? That might be easy, but it's hard. I've never been able to see a neural mechanism for that was easy. Whereas in the 1D sense, it was pretty easy. I only had to have as a scalar value per 1D mini column. And that scalar value would be basically sort of represent the how orthogonal the 1D dimension is relative to the orientation. It's sort of like, it's how much is, if I'm moving forward, how quickly does this 1D module change versus other ones?
Right, it's just a projection onto 1D, and you use that to update some scalar value, which you could do with 20, 30 cells.
Or even less than that, because I was arguing that could be actually, and also I figured out a way of using sensory input to determine that. And therefore, what I was thinking was, if you had, let's say, a bunch of bipolar cells that were representing orientation, and those bipolar cells received input from sensors that were flow information. It wasn't a spatial pattern; it was a directional flow. So the cortex is infused with cells that are directionally sensitive, and a directionally sensitive cell would actually represent, it would represent the sort of the projection of orientation onto that sensor. In some sense, I could use direction-oriented input from an eye or from a finger and put them into a sort of spatial Pooler process. And what I get out of it, I get a bunch of scalar values that represent the projection of orientation onto that dimension. And so I could, using a simple spatial Pooler process, using sensory data I know exists, I can now have a scalar value in a bunch of mini columns, and that scalar value could just say, if I'm moving, how much should this bump move? How quickly should this bump move?
But it's going to be, it'll only work for moving forward.
Yes.
If you move some other direction.
Well, if you move another direction, then you have to change your orientation. And as soon as you change your orientation,
different cells are going to.
Yeah. So.
What? Moving some other direction requires moving.
Yeah, of course. I mean, that's what's all over visual cortex. All the orientation selective cells. They're actually different.
But just start with the simplest explanation that a rat either can be training its orientation or it can be moving forward.
So it can't step left and right. It can. Okay. Oh, it can't walk. Okay.
Yes. Let's take that for a start.
Okay.
I realize maybe the rat can do I can do that, but let's just start for the idea that the moment, and maybe if I'm stepping sideways, maybe what I'm really doing is changing my orientation, and I'm changing the direction of my head. So if you think of orientation as the direction you're going to move, not which way your head is facing, it might work. But yes, I agree. I'm making a bunch of simplifications.
It's like my motion in time periscope last week.
So yes, so the idea that, and then here too, as I was going, another thing I think nice about this is if I think about this, I think it's a thetaism between it's theta between the cortex and thalamus, and I've argued in the past that might be a scalar that represents the speed of movement. And so then all you needed to have an orientation and a speed of movement, and then all the individual one-dimensional cells would update automatically. So at least I could walk through a scenario, and I think I could get it to work, where I'm using sensory data to determine my orientation, and then I just need a scalar in each mini column. And now when I move, I know how to update. I can use that scalar to scale the movement of the box, and I could get a system to work.
But I could very easily get that to loop to get all the oscillations in the 1D that you wanted in terms of a periodic frequency of the system.
You're just saying that the grid cells repeat? Or?
Yeah. You just provide additional input and it loops around.
So the problem here is that, again, as I started off last week, as I said, well, the empirical evidence doesn't suggest it's 1D. Empirical evidence suggests it's 2D. Right. But anyway, going back to your point there, your question, Marcus, I felt like for the first time I had a workable system where I could say sensory input can determine my orientation, it can tell me what the number of dimensions I have in the world. It can tell me how to update my grid cell modules. I have enough grid cell modules that I could sample from to get the right answer. And even I had enough grid cell modules that I could use a sort of higher level encoding, which is making some look missed. Basically, like let's say I had 200 grid cell modules, and they were divided into 3 dimensions, or 3 60-degree dimensions, or something like that. Then I could activate some subset of them to represent. They could all be updating, but activate some subset of them to represent the state of the system.
You get combinatorial complexity on top of your 2D lattice that you're spanning with those 60-degree.
Yeah. So I don't know. I made it very clear that it wasn't a full. It didn't fit everything, but it was an interesting thing to think about. It was like there was something about, and I really like the idea that I could use this sort of flow of sensory data, like the directional sensitivity, to know how to update my orientation and my position.
Yeah, so that I still need help with. So I feel like you're changing the philosophy of what an orientation is a little bit. For the most part, up until now, an orientation doesn't depend on sensory input. Direction cells don't care what you see.
Well, we did, but we generally don't talk about, but remember, we did talk about this in the past. Remember when we talked about playing a video game, and you're sitting in front of a screen, and you're playing a video game, and you're moving your character around in it. And we know that's one of the scenarios that the Konstantinescu people did to test their theory, right? And what we know is even though you're not moving your body, you have a sense of moving through the space and your location in that space. And so
fine, but you turn left, and whether you see Mario or Luigi doesn't influence.
No, but the whole room shifts left, right? So again, remember, it's the flow that matters. So, so where I went with this,
a change in orientation, yes. The deltas to your orientation you can get from sensory input.
What I'm saying is the delta. It's imagine the following. Imagine your sensory data is divided into 2 parts. One is a spatial pattern, like the impressions on my finger and whether I'm detecting an edge or not. And the other is the sensors that detect motion across, and independent of the pattern. Those exist, I believe, in vision and in touch. You have these 2 separate things, 2 separate pathways. Now imagine if I take the pathway that's representing flow. I can't, or I don't know if I term for it, movement. I can't determine what the actual thing what's the pattern of my finger. I can just tell which way it's flowing. If I take that information, now I have the ability, imagine now I'm looking at a screen in a game, and I'm like, how do I know I'm turning left or turning right or going forward and going back? The only information I have by looking at the screen is the relative flows. That's it. There's nothing else matters. It doesn't matter what objects are there. It's just the relative flows. So one set of sensory data can determine, and we decided this once before, that the cortex has to learn. We talk about grid cells being updated by motor behavior, but it actually has to learn using sensory data. We concluded this once, that had to learn how to use sensory data to first determine your movement, and then you could associate that with motor behavior. That up front, you don't know what motor behavior is making you move. So it really had to be a sensory-driven experience, and the video game is proof of that. It tells you that I can just watch the screen, and I know which way I'm moving, I know which way I'm turning, I'm going forward, going backwards. All this comes from a sensory data, and I think the same could be arguments for your fingers. That's not normally how we talk about it, but we did talk about that in the past once. we came to that conclusion. That, in fact, I believe that is the primary method by which the cortex learns, uses to detect movement, either change of orientation or moving forward or backwards. And later it associates that with a motor behavior.
Yeah, so I totally agree. It's but the key thing it's movements, it's changes in orientation. It's not the orientation itself. It's not the location itself.
No, you're asking how does it get anchored? Is that what you're saying? How does the orientation get anchored?
I think he's saying absolute orientation versus relative, even that's independent of the
so
anchoring
that's
argument.
anchoring, right? That's just basically, that's relative orientation. Now I have to anchor it to some environment.
Right. So if you drop something down, and it just wakes on up and it's not moving, can it determine what its orientation is? If you don't have it.
So the basic idea we've talked about in the past is obviously now you wake up in some you're blind, you open your eyes, or you're just started in some environment. You don't know where you are. You don't know what your orientation is. You have to infer that. You just can't know it a priori. You have to
right, but you don't have to be necessarily blind. Just you wake up and you can't move. Can you determine an orientation? Well, the answer is if you're learned, if you have some priors, you can do that.
My point was you were blind up to the point you opened your eyes. That's all I meant by that, Kevin.
Okay.
You don't know where you are. All of a sudden, you have sensory input, and you have to sort of figure out where am I, which way am I facing.
Right.
And that requires being in. If it's in a learned environment, you have to be able to recognize something in that environment. So the idea there is okay. I have some sensory input. I see something in front of me. If I recognize that thing then I might know where I am and which way I'm facing.
So given that, the question is, does it require a grid cell system to give you that recognition, or is that an independent system? The grid cell, it's a question of whether the grid cell is fundamental to orientation or whether it is an auxiliary system that helps you with other inputs.
They go together. You have a model if you've learned this environment, you have a model of the world, and now you open your eyes, you have to infer both your location and your orientation. And you can only do that if you recognize something in front of you. So but it's the interaction between the sensory input. This is the spatial sensory input.
Right. So another hypothesis could be that the grid cell only gives you that information under the condition of deltas or movement, that you can infer it cognitively from something else. So that's not
I didn't follow that, Kevin.
The question is whether the grid cell's ability to detect orientation. I'm just trying to go with what
grid cells
saying.
Don't detect orientation. Grid cells are orientation independent as far as we know.
No, I think Kevin's point is the idea. If you were to teleport the rat from one spot to the next, would the grid cell still update?
No. How would it possibly know what to do? It can't. Physically impossible.
Well, I mean, based on sensory data. And
okay. So based on sensory data, right? So when you say teleport, I assume there was a blanking interval between the time A and time B. Right. If I move the rat, it's like me looking at a screen, and all of a sudden my character in a role game just starts moving forward. Yes, I would infer where I am.
Then you have to. I mean, there's clearly a motor component and the.
That's purely a sensory component. I'm not doing any motor output. Right, I'm just looking at a screen, and the first person perspective of my character is moving through the screen. There's no motor output on my part. I'm just inferring that movement through visual input. That was the key insight of thinking about video games.
What about proprioception?
I'm not even moving. I'm frozen. I'm just looking at the screen.
Okay, right. In the video game case, of course, yes. We don't have walking controllers. I was more thinking about the rodent.
No, but I'm using so this was a big twist in my thinking a long time ago. We always thought about grid cells as being updated by motor. So that's the only way they're going to be updated. You have some motor behavior, or proprioceptive input, whatever. But then we realized that there was a problem with that. And one of the problems is we don't know how to learn that association between motor and the grid cells. The brain doesn't know this up front. It doesn't know what body you have. So, then we realized with the video game example that proves that all that can be done purely by visual input or purely by sensory input. And then it becomes, if you're detecting flow, movement. And then it says, okay, maybe that's the primary method by which we do this, because I know that I can do it independent of any actual physical movement. And then you can use that to associate with the motor behavior. So that's where I'm coming at from this. So I don't know, that's something that I felt, again, one of the things that's nice about this, at least the way I understood it with the 1D cells, is I can imagine how sensory input that's detecting movement or flow can be put through a spatial Pooler process, creating a bunch of mini columns that then represent 1D dimensions of space, and can update appropriately based on that, the output of that spatial Pooler. And then when I'm going back in a new environment, I have to infer it again, right? Yes, I have to re-anchor. I have to figure out where I am and re-anchor. And I don't, Marcus, I was along. Did I answer your question, like what it is I thought this was solving that I couldn't solve another way?
I'm still a little confused, but I don't think the right thing is to. I think I should stare at the board a little longer.
Maybe you and I could talk about it prior to you one. I can just try to walk it through for my benefit and see if I can make it work. Sometimes, maybe I'm fooling myself and it won't work, but it feels like it should work.
Playing more times always helps make it more.
Yeah.
Different listeners, different perspectives.
I just felt for a long time I'm sort of stuck on the whole grid cell thing. There's so many things that just don't add up. We don't have enough modules. It's not clear how to divide up or n-dimensional space. We don't know how to learn that re-anchor. We don't know how to learn what the dimensionality of space is. Do we even learn the dimensionality of space? Right. How is orientation represented in a 3D world, such that I update my grid cell modules in a 3D world.
Right. And then higher order cortex, where those metrics would no longer be spatial, but cognitive dimensions. How would you build that, relevant space? How would you span it, given the resources that you have?
That's hard to imagine because, like the bird space. It's like, I can imagine how I could model orientation.
The Constantinescu paper.
Yeah,
right.
Yeah. At least I can imagine how sensory input that, if I divide it into spatial patterns and flow patterns, how I could do all this. But I don't know what the equivalent would be in bird space. What is the flow in bird space? I don't know what that is. I even had this idea, just totally speculative, just throw it out so Marcus can hear it and think about it. I had this idea that, okay, maybe there were you always talk about the supergranular and infragranular layers. And I said, what if we had a set of the mini columns in the supergranular layers are driven by the spatial sensory input, like we've always talked about it, like a spatial pattern. And so it's like a feature or something that detecting what the object is. And what if there was a separate input coming in layer 5, which we know there's a separate input coming in layer 5, which represented the flow sensory data. And from those mini columns there on down, that they're all representing space. And so I have the space analysis, the mini columns representing space in the bottom, and the mini columns representing object features in the top upper areas, and how those, because that's basically the place cell, grid cell bipolar. And one testable hypothesis is this: we know that there's these two sensory inputs that come from the thalamus to cortex. One goes to layer 5, and one goes to layer 4, and nowhere layer 3. And this has been over and over again. But if the inputs to layer 5 could be shown to be specifically ones that are related to movement in the sensory stream, and that's known to exist, that they're there, I don't know where they project, that would support this hypothesis. So basically, if you have two different sensors on your eyes, ones that are spatial patterns and ones that are detecting movement, that the movement detectors would go into the lower layers and the spatial patterns would go into the upper layers, that would kind of fit with this model too. And that's something that might be known. That data may already be out there. I would give it a more than 50% chance that people know that. I didn't look it up.
I think that the second thalamic input is more to 6A, maybe lower layer 5, but the thalamus projects to 4 and 6A primarily.
Does it? I thought there was a strong input to layer 5. I thought it was lower layer 5 and maybe upper layer 6. Could that be?
That's possible.
That's possible.
But like, for example, this is possibly why so-called simple cells are in 4 and 6A.
Yeah.
And they're the ones that are receiving the direct.
Oh, it's interesting. I think that, this whole idea that, I guess I'm sort of questioning the whole idea that, that the same idea of what a simple cell is across all different layers is maybe wrong. and maybe in a situation where you have an anesthetized animal and you're just giving it, well, think about how, you know, what happened with Hubel and Wiesel. They couldn't get any cells to respond to anything until they accidentally slid the slide in, right? And so the edge of the slide moved across the visual field of the cat, and that was the first time they ever recorded a cell firing based on the input they get. They were trying to show the cat all these inputs. So now what do? Now when we want to characterize what a cell responds to, we show it sinusoidal gradient, which is almost all movement. It's not really patterns at all. Right. It's just a movement vector. And so again, that's the idea again, that maybe the activity of a column is really representing. By the way, that movement is literally, if I show a sinusoidal gradient and cells, we talk about different orientations, and they respond a graded response based on the orientation. That literally would represent the projection of the orientation to the, in some sense, the flow would be defining which is the primary 1D dimension, and other columns would respond gradedly or not at all, depending on their projection onto that. So that whole idea of what a mini column is representing an orientation column, that's almost always determined by movement, like flow through sinusoidal gratings, which that really what it's detecting is the response to the mini column then becomes less of the pattern, it becomes more the orthogonality of the projection of the orientation to that movement. Like it says how much is this mini column represent movement in that direction is what it represents. That became the signal that we could use to update the location cells. I think there's something to that idea. It just that fits. I don't know. I'm frustrated by this. I'm talking too much.
Did anyone ever do an experiment by, instead of dragging gratings across, dragging random dot patterns in a particular direction across?
I don't think it would matter. I think the point is it doesn't matter what the pattern is. It just matters that it's flowing in a particular direction.
Well, that's the interesting thing is whether, you know, I mean, theoretically, there are cells in the LGN that respond to certain spatial frequencies over others. So I'm just looking at, you know, if you have a composite thing where it's made of, you know, a huge number of spatial frequencies, whether there's some place where that all correlates and saying, yeah, I still got motion irrespective of, you know, I mean, do all the grading patterns light up, you know, when you drag random dots across?
So what's your point, Kevin? I'm missing it, sorry.
My point is you had mentioned that until they dragged an edge across, it didn't get any response. What I'm wondering is that if anyone's tried it where it's movement of the textures rather than a specific spatial frequency grid moving across, and whether that correlates and does that create the same firing pattern, if you wish, in where you're looking for response.
They tested those things if you go back into the 60s and 70s.
Okay.
I don't know, but I'd be surprised if they didn't try different things, because they didn't start off thinking about sinusoidal gratings. They tried, you know, again, the edge of the slide, you know.
Right. I'm just wondering if you know, with the better experimental technique right now, whether that would actually help kind of resolve, you know, at some level where some of this is occurring.
I'm not sure how that would. I mean,
Okay.
we showed it other patterns. I think they chose the sinusoidal gratings because they're the most reliable.
Mm-hmm.
Those were the ones that
contrast.
It was the one that the quickest. Remember, in the end, they want to characterize these cells. Usually, there's some experiment they want to like figure out what they quote the classic receptive field for the cell is. And so they quickly came out decide that the best way of figuring that out, the best responses they got were from sinusoidal gratings. They didn't start there. So I don't know what else they tried and what the responses were. Obviously, the moving edge of the glass slide worked for some cells.
Yeah, well, given that you have, if you wish, feature detectors in the optical cortex for actually finding edges, that makes sense. I was just looking at, you know, where what the Gestalt sense of motion, where that would locate, whether it's just an integration of all the spatial cells, or whether
I think that it's known that there are motion sensitive cells in the retina, and there are motion sensitive ganglion cells. I think this, I have to go back and check this. I know there's motion sensitive cells in the retina. I think they're preserved. I even think they're kept separate as I go through the thalamus. But I don't know if what's known about where they project.
Yeah.
There clearly are, and Marcus had done more recent reading on this, there clearly are motion detective sensitive cells in the skin. I know there are motion sensitive cells in the auditory cortex and the cochlea because they detect, moving frequencies. In fact, it's almost so much of, if you go look at when they characterize these cells in the mini column in like V1s, almost all except a vast majority, if not all, but many of them are motion sensitive. And they only respond, or they respond preferentially when the stimulus moves in one direction or the other direction. So it seems to me like we've kind of forgotten that this motion sensitivity is sort of inherent in the mini column. It's a key criteria across every mini column ever been looked at.
Okay.
And I'm just saying, here's an explanation for it. It could be a way of determining the orientation of a mini column in a space. A relative orientation, because obviously different mini columns will be getting different activations. All right, well, this is good data. Thanks, Florian. It suggests that there's not a simple explanation that there are 1D cells, at least in the entorhinal cortex.
Yep. Exactly.
But I'm not giving up on it yet.
No, there's no reason to give up on the idea.
We don't have a working hypothesis we can go back to. We have a non-working hypothesis. So we still have a non-working hypothesis.
Okay, guys, I'm gonna go offline now.
Thanks.
You might want to move the double since I'm in the way. I know.
Cool.
Okay. So, you guys hear me? Okay. Good. So thanks for watching. A couple things before I let you guys go. That was a long research meeting, and I know we got off late with it and everything. Whoa. I have to turn off my meeting just a moment. End the meeting. Okay. Great. Everything's good. Okay. So if you're watching this, you might be interested in, now I feel totally like I'm not prepared. Here it is. Over to this. On December 23rd, that's when Gary Marcus and Yoshua Benjuo are debating. And so I thought we would do a live stream. People do this on Twitch sometimes. You watch something live with your audience. So I'm inviting all of you guys out there to come join me December 23rd. That's a week from today at 4 p.m. Pacific, 7 p.m. Eastern on my Twitch channel. And so that my Twitch channel, for those of you not familiar with Twitch, it can be sort of confusing. This is my Twitch channel. Somebody else is on it right now. That's fine. That happens. But if you go to this Twitch channel, I don't know what any of this stuff is. This is an ad or something. But if you go to my Twitch channel in 1 week at 4 p.m. On Monday, you'll get to watch me watch Gary Marcus and Yoshua Benjuo debate live for as long as they go. And then we'll chat. I'll have the chat room open. And we will also have I'm thinking some type of polling or voting mechanism so that you guys can interact, give people points when they deserve them. I don't know. We'll do something fun like that. I'll have something figured out by then. And I'll be prepping for it on my Twitch stream if you want to check out my Twitch stream. Please like this video and subscribe to the YouTube channel. It's very helpful. I know it seems silly. Everybody asks for it. That's because everybody wants it. And also the very last thing I could mention is I got a new HTM School video, in case you missed it. HTM School 16. It's about hierarchy. There we go. Thousand Brains Theory and Hierarchy. So I really explain hierarchy. Just Google it. I explain why hierarchy in the brain is different than the way we think hierarchy in the brain, or excuse me, hierarchy in deep learning networks work. So anyhow, thanks for watching. I am kicking off now. I gotta find the right button to turn all of this off. Take care, everybody in chat. Oh, I think people are chatting, aren't you? There you are. Yeah, bye everybody. Bye Falco and Coraline, Chris, Eric, John, and Allie. There's a bunch of you guys chatting. I appreciate you coming in and joining our live streams. Please share our research with everybody out there. Have a wonderful week.